started, and as people file in, I'll just catch up on the conversation.
Sure.
Welcome everyone to the first Journal Club of 2022. So just a quick background on the Mass Journal Club itself. We're a monthly initiative for the academic discussion of psychedelics, and it's started by students from the University of Toronto, McMaster University, and Queen's University as well. So for today, we have a talk by Dr. Ju Liu from the University of California, Santa Cruz on an analog of psychedelics that restores functional neural circuits disrupted by unpredictable stress. So Dr. Liu has a interdisciplinary background in neuroscience and engineering, and he seeks to understand how the brain processes information and how such functions are modulated by experiences and affected by diseases. After studying microelectronics as an undergraduate at Tsinghua University, he received a Ph.D. training in neurobiology at Harvard, during which he reconstructed the first mammalian neuromuscular connectome. He then did a postdoc at Stanford University, working on super resolution microscopy and deep brain imaging. And currently he is an associate project scientist at the University of California, Santa Cruz. And so with that, Dr. Liu, could you please take over with the screen share?
Okay, all right. So hello, everyone. So first of all, I would like to thank the organizers of this journal club for giving me this opportunity to share my recent work with you. So as we all know, life is really full of stressful events. And we have all experienced the kind of social isolation imposed by the pandemic. And the stress can also come from things like, financial difficulties or family crisis or work pressure, or like natural disasters like these wildfire that scorched Northern California 2 years ago. And also, the kind of political turmoil that we have all observed previously. And for researchers, of course, these unfavorable opinions from journal editors. And in order to study how stress affects the brain and to figure out ways to treat or prevent it, I'm adopting a mouse model, and particularly I'm using this so-called unpredictable mild stress model on the mouse. So in this model, the mouse is subjected to a number of different mild stressors on a rotating kind of randomized schedule. For example, it may be exposed to light during its normal dark cycle of the day, or it may be temporarily deprived of food for like 12 hours, or it could be stuck into a tube so that it is kind of very confined there, can't move around for like an hour, or it's intermingled with some stranger animals from another cage, or isolated, or exposed to some loud noises and so on. And after a week of such unpredictable stress, we can test how its behavior and how its brain circuits are altered. And before we go into the details, I would like to remind you that the mammalian brain is a very complex network with a hierarchical organization. So this is a mouse brain. And if we cut it up, take a cross-section, we can see that it consists of many different regions. And each region has its specific cell types, its connectivity with other brain regions and specific functions. And many of these regions, such as the VTA, the ventral tegmental area, and the NAC, the nucleus accumbens, they're well known to be involved in like reward and reinforcement learning. Amygdala is important in like fear. PFC, the prefrontal cortex, contributes to like higher order cognitive functions. The hippocampus is important for spatial navigation and memory. And all these regions have been quite extensively studied in the context of stress. However, the primary sensory areas, such as the somatosensory cortex here marked as S1, those areas have received much less attention among stress researchers. But if you think about it, the sensory cortices are the early stages where external information enters the brain, and then they are processed there and then feed forward to other brain areas. So if things go wrong there, you would expect that emotion and other more complex decision-making would be interrupted because they are based on a kind of erroneous input signal about the environment. So that's the region that I'm focusing on in my study. And if we zoom in to the primary sensory cortex, we can see that the cortex is organized in different layers, as marked here, 1, 2, 3 through 6. And here you can see individual neurons labeled by a classical staining technique called the Golgi staining. So they appear very dark, and you can see their processes extending from the cell bodies up. And here we only stained a very small fraction, about like 1% of the cells in the brain. So you can actually see them individually. If it is all, the entire brain, like all the cells are stained, then it's basically not resolvable. And if we zoom further in to part of this cell, we can see structures like the dendrite, which is marked here in green. So this is the input cable of this neuron or the nerve cell. And the yellow ones are the axons. These are the output cables of another cell. So as you can see here, the axon touches one of these little protrusions from dendrite. These protrusions are called the dendritic spines. And they form a connection where information is transmitted from one cell to another. So as you can imagine, if you have changes in these dendritic spines, that essentially reflects a change in how the neural network is wired up. And if we can examine these changes in the dendritic spines, we can infer how the neural network is responding to experiences such as stress or learning or diseases. Okay. So previous studies have established that stress really disrupts this so-called synaptic circuit in the cortex. So as shown in this cartoon, after stress, these arborization of the dendrites, these incoming input cables to a cell, get reduced. And also these protrusions on the dendrites, the dendritic spines, become fewer. So that suggests that this neural circuit is really disrupted. So is there any way to correct the kind of detrimental effects imposed by stress? Well, so here the psychedelic drugs come into play. I guess I don't really need to dwell on how marvelous these drugs are to this audience. I just want to emphasize that it is kind of established that these classical psychedelic drugs, such as the LSD, the psilocybin derived from magic mushroom, or DMT, they all work on this class of receptors in the brain called the serotonin receptors. And the previous studies, both on animal models and on human subjects, have shown that these drugs have a potential in treating a variety of neuropsychiatric disorders, such as addiction, anxiety, and depression. However, because these drugs generate hallucinations, it is a concern for clinical use. And in order to get some drugs that retain the beneficial properties of the classical psychedelics, but do not generate hallucinations, my collaborator, Dr. David Olson at University of California, Davis, did a synthetic work to derive from ibogaine, which is a psychedelic derived from this West African shrub, tabernanthalog iboga. So he changed the chemical structure of ibogaine into a novel drug called the tabernanthalog, or TBG. And the TBG possesses a number of desirable properties. So it is soluble, so it is easy to synthesize, and it doesn't generate hallucination. And in the earlier work, we also showed that the TBG has low cardiotoxicity, which is actually a problem for ibogaine. And moreover, it promotes the growth of the dendrites and dendritic spine formation in the wild type animals. So that gives us the hope that it may be a good candidate to correct the neural circuit that is disrupted by stress. So in order to evaluate its effect on stressed animals, so we first did a bunch of behavioral tests. So here I examined the anxiety level of the mouse with this the elevated plus maze test, which I will go into in a second. And we also tested its sensory processing with texture discrimination test. And finally, the animal's cognitive flexibility with a 4-choice odor discrimination test. So basically, the experiment goes like this. So we stress the animal for 7 days, then give it a shot of TBG, wait for 1 day, and do the behavior test. So first, in this elevated plus maze test, we have maze, which is basically in a cross shape. The closed arm has a wall, so it is safe for the mouse to stay in there. The open arms do not have walls, and the whole thing is elevated from the floor by some posts underneath. So it's about like 50 centimeters away from the floor. And normally, the animal would be kind of curious to explore these open arms, but at the same time, it has an innate fear of height. So it is a trade-off between this curiosity and the fear. And as we can see in a control animal, it does explore the open arms during its time on this maze. But after stress, the exploration of the open arms reduces. So that suggests the animal has a heightened level of anxiety. And then after the TBG treatment, the exploration returns basically to the control level. The second test we did was the cognitive flexibility. So here, the idea is that we first get the animal to learn which odor is associated with food reward. So we place it in the arena, and at each corner, we have a container with the distinct odor. So we just put some essential oil, like different kind of essential oil, on a piece of filter paper and attach a paper to the container. And then we put a piece of breakfast cereal at the bottom of only one container and cover it with some wood shavings. Each container has the same amount of wood shavings, but only one has the food buried underneath. So the mouse will have to learn to dig into the wood shavings to recover the food. And while it does so, it will gradually establish this association between the particular odor and the food. And we measure its learning by like how many times it has to try before it finally can do the digging correctly with like 8 correct trials in a row. So that's the first phase. The second phase is that now we change the rule of the game. So the food is associated with a different odor that previously had no food reward. And now the mouse has to relearn this new rule and see how many trials it has to undergo. So as you can see in the control animals, it actually learns pretty fast. But after stress, it learns much slower, which suggests that it has a more rigid kind of way of thinking, right? The initial learning actually doesn't get worse, but once it learned something, it is very difficult for animal to change its mind to learn a new rule. And then after the TBG treatment, the animal can again have this cognitive flexibility for the reversal learning. And the third behavior test is on the animal's ability to discriminate between two textures. So here I'm showing a video. Basically, the animal is placed in this arena, and here are the two wooden posts wrapped with sandpaper. So initially, it is one kind of sandpaper, two same textures. The animal just runs around and gets exploring the textures. Then after 5 minutes rest, I would replace one texture with a novel texture. And the other one would still be the same texture that the animal has become familiarized with. Then, because the animal innately has a preference for novelty, it would interact more with the novel texture than the habituated texture, as shown here. So here I'm calculating this so-called discrimination index, essentially as the difference between how many times it visits the novel texture minus the time it visits habituated texture and divided by total number of approaches. So it has a significant preference for the novel one. But after stress, as you can see, the animal completely lost this discrimination ability. So it would visit both textures equally. And this texture discrimination ability is restored after the TBG treatment. By the way, before the testing phase, during this encoding phase, when there are two identical textures, animals never show any preference to one side or the other, which is good, right? Which suggests that they are not preferring one side just because there are some other cues that prompt them to do so. So having established that the behaviorally TBG can rescue the impairment done by stress, I went on to examine how TBG work on the brain circuit. And in order to do that, I leveraged the technique called the two-photon microscopy. So normally, when you look at fluorescent sample, for example, you shine light on it, and that light excites some fluorescence emission from the fluorophores, and then you can observe the fluorescence. But in that case, you essentially on the molecular level, you have one photon coming in, and then it's absorbed by the molecule, and then one photon comes out. But in two-photon microscopes, it uses a very powerful infrared light from a special kind of laser. So the fluorophore needs to absorb two photons at the same time to emit one single fluorescent photon. And that gives these two-photon microscopes some unique abilities to image deep into the brain, and also to have this so-called optical sectioning ability, as I shall show you in a moment. And in order to image into the brain of the mouse, we can either just very gently thin down the skull of the mouse, but without penetrating it. You just thin it down to the point where the remaining skull is only like 20 to 30 microns thick, and it becomes basically transparent. Then you can image through the skull to see into the brain. And here, what we are seeing is like images taking progressively deeper into the brain And you can see there's basically no autofocus light, right? You can see these processes very clearly. And the processes are actually labeled by expressing a fluorescent protein genetically in the cell. So the protein will fill up the whole cell and make them visible under the microscope. Alternatively, you can make a window through the skull and seal it with glass. So here you are seeing such a window, and you are seeing the blood vessels on the surface of the brain. And then we can label the cells by either transgenic techniques or by injecting a virus. And then these cells shown here are expressing a special sensor for calcium. And that sensor will indicate when these cells are active. So here you are seeing these individual cells, and some of them would suddenly light up, like here and here, and here, for example. So by doing this, we can actually see how the individual brain cells are activated when the animal is doing some particular task. So first, I was looking at the structural changes. As I mentioned earlier, these dendritic spines are really representing the neural circuit wiring. So as you can see here, I first image the dendritic segment on the day before the animal undergoes stress. And then after 7 days, when the animal finishes the stress protocol, I image the same dendrite again. And you can see a bunch of these spines, these little protrusions are disappearing. Like this one is no longer there, right? This one is no longer there. This one is missing from here, and so on. So there's actually a pretty substantial loss of dendritic spines. There are also some growth, like this guy, but they are much fewer than the loss. And after a day of recovery, the situation doesn't really change much. But with the TBG treatment, as you can see here, still, the animal undergoes the same kind of stress protocol. So there's also spine loss shown here, here, and here. But with the treatment, within a day, you see a lot of new spines being formed. And if you do the statistics, indeed the formation level is much higher than in the control animals or in just the stressed animals after spontaneous recovery. And more interestingly, if you look at these newly formed spines, about 20% of them form at exactly the same sites where previously a spine was lost. So that means like this drug is really at least partially restoring the neural connections that were previously lost due to the stress. And that, I think, partially explains why this drug can restore the behavior, because the neural circuit is kind of repaired. And then moving from the structural observations to more like a functional investigations. So we first did a mesoscopic calcium imaging over the entire window. So here here's an example. Well, you can see this animal is awake and sits just on a disc, so it cannot move its head, but it can run if it wants to. And at the same time, we can image what's going on in its brain and monitor when it is whisking, because here we are actually looking at the brain area that is responsible for detecting the signal from its whiskers. So here you can see part of this lights up. That is the neuronal activities in this area. I mean, here we don't have single cell resolution, but so this represents a relatively large group of neurons. But if we correlate the whisking with the brain activity, we can see they have a pretty high correlation, right? When the whisking magnitude is high, you also see an increase in the fluorescent changes. And this correlation is actually greatly diminished after stress. But with the drug treatment, it is like restored to a large extent. And moreover, if we line up the onset of each episode of whisking and see, how the brain activity looks like when the animal starts whisking, you can see in the control animal, there's a very clear onset of whisking associated brain activities, right? Before that, it's pretty quiet. After the whisking starts, the brain activity goes up. But in stressed animals, after whisking onset, it is still, about the same level of activity as before. But before that, you see a lot of these activities. So that suggests that there's some sort of signal to noise problem, right? The brain is like active when it really shouldn't be. And that could mean like it is detecting some wrong signals and transmitting it to like other higher order brain regions. But after the TBG treatment, it becomes clean again, right? Before the whisking onset, there's not that much baseline noise anymore. And then we move on to the to monitor the cortical neural activities while the animal actually performs this texture discrimination task. So here we have so-called neurotar device as shown here. So the animal is head fixed, but it can still move its limbs. And this arena is made of carbon fiber. It's very light. And then you have air coming from all these little holes on the base plate. So this arena is essentially airlifted. So when the animal moves, the arena moves in the opposite direction. So the animal can still approach the textures attached to the side of the wall, as you can see here. And at the same time, we can place these underneath the two-photon microscope. And now we can really image individual neurons' activities. As you can see here, they are like flickering, right? So here, each of these is a single neuron, and we can really monitor their activities while the animal is doing this task. So here is, I'm showing you some examples of the activity traces of separate neurons. So each trace represents that of one neuron. And here we can mark, when the animal is interacting with the novel texture, when the animal is interacting with the familiar texture. And then we can examine for every neuron what it is doing. Is it preferring to be active when the animal interacts with the novel texture or with the familiar texture, or the neuron actually fires indiscriminately whatever the animal is doing, right? So we actually find different subgroups of neurons. So here we call these novel selective neurons. So if you line up the time to the onset of interaction and see their fluorescence changes, they become bright when the animal starts to interact with the novel texture, right? So that means they are active when the animal is interacting, but they are not active when the animal interacts with the familiar texture. And conversely, there are neurons that are active when the animal interacts with the familiar texture, but not so with the novel texture interaction. Then there are neurons that are just firing indiscriminately. But if we look at the percentage we have about a third of neurons that are novelty selective. We have a much smaller fraction that are familiar texture selective. And, half, a little more than half of them are not responsive to this. But after stress, as you can see, these texture-specific neurons almost disappear. You only have very small fraction of cells that are still selectively responding to one type of texture. And that probably underlies the fact that the animal is no longer discriminating between the two textures. Now, after drug treatment, these two populations are kind of partially restored. Not completely restored, but still to a substantial extent. Right, as shown here. It's basically abolished the novelty responsive neurons. And then after drug treatment, they are coming back. So in summary, we found that stress can induce a number of behavioral deficits in terms of affective, sensory, and cognitive functions. It can disrupt the cortical neural circuit as well as the function of cortical neurons. And a single dose of the tabernanthalog or TBG can promote the regrowth of synapses and rescue these behavioral and the neural activity deficits. So its rescuing effect actually highlights the therapeutic potential of psychedelic drugs analogs in treating these stress-related mental disorders. And finally, I would like to thank Dr. Yi Zhou, my supervisor, as well as my colleagues in the Zhou lab at UC Santa Cruz, my collaborator, Dr. David Olson, Dr. James Ackman, and Dr. Lu Chen. So they have all contributed to different aspects of this work. And thank you all for your attention, and I'm happy to take any questions.
Thank you so much, Dr. Liu. That was such a great way to start off the new year. It was a great, super interesting talk. So just a reminder that you can enter your questions in the chat, or if you want to raise your hand. So we'll start off with Arseni, who has hand raised. Leo, could you just unmute, please? I don't have the ability to.
Hi, can you hear me?
Hello, yes.
Oh yeah, perfect. Yeah, I mean, there were some questions in the chat, but if you don't mind, I actually had several questions. So I will start, I just started with more molecular one. I'm not really familiar with the TPG, but I was wondering if you checked or if you know what exact receptors it binds to, like, because we know that psychedelics bind to serotonin receptors and 5-HT2A, 5-HT1A, which are implicated in stress. So I was wondering if you checked for the binding sites of the
yeah, that's a great question. So as you mentioned, all the classical psychedelics are agonists for the 5-HT2A receptors, right? And TBG is also an agonist for 5-HT2A, but in the previous paper, in that Nature paper published last year, which describes the synthesis and pharmacological characterization of TBG, Dr. Olson's lab actually did screening of the binding partners of TBG. So it is not a very clean drug, if you will. So it, in addition to 5-HT2A, it also has affinity for 5-HT2C. It has affinity for some other receptors to different extent, just as most other psychedelic drugs are. So we know that its effect on the behavior requires the activation of 5-HT2A. So if you block 5-HT2A with an antagonist, then you do not see its effect. But we cannot say that the drug's effect is exclusively mediated through 5-HT2A. So that, I think, requires more studies to really tease out the underlying molecular signaling mechanisms.
Sorry, Leo. Yeah, I think just the way the settings are, we got to just unmute manually each time.
Okay, I got it.
Yeah, so yeah, thank you. So the other question I had is about the timing, because there's a delay. I wasn't sure what was the delay after the stress that you gave the drug, and the testing after you gave the drug. What was the delays there?
Okay, so let me show you a timeline here. So usually we do this. So we give the drug immediately after the stress protocol ends. And then we wait 24 hours before we do the behavior assessment.
So I had like two questions. One, did you try to see how, like, if you, for example, tried it 2 days later or 3 days later, did you see the effect? And if you tried it before the stress, would it have the same effect as if you gave it after? So like, do you know?
Yeah, that's very good question. We haven't done that. I think that's something we will do, like to see whether it has a preventative effect, and as well as to see if it has a long-lasting effect. I think it probably has a long-lasting effect because there are some other studies on like a psilocybin, for example, which suggests that the effect on the synaptic circuit seem to last at least like a week or two after the drug administration. And I expect that TBG would have a similar effect.
Thank you. Yeah, and yeah, I think there was a question in the chat, so I'm gonna just give it because I was really interested in this that you said 20% of like the spine growth. That was really interesting because I was wondering as well, like how, because there should be like marker molecules in that case that would like tag those sites, and then but like there's a question in the chat, so I'm just gonna give it to people in the chat. Yeah, thank you much for your answers, and thank you so much for the talk.
Thank you.
Perfect. Yeah, thanks for the great questions there. So yeah, one of the questions in the chat was talking about the spine regrowth. And so they're saying, so they regrow spines, but if the mice were not stressed, does it accelerate the spinal formation faster than the normal formation rate?
It does. It does enhance the spine formation rate a bit. So that was actually reported in the original paper describing the TBG.
Perfect, thank you.
And actually, you know, some other psychedelic drugs also have this pro neuroplasticity effect.
Perfect. Another question was, so was there any variability in how the animals responded, either behaviorally or even just with the effects you saw with like the spine regrowth, for example? Like, did you see, was it pretty uniform within how the animals responded, or were you able to see some variability there as well?
Well, I mean, here you can see the, here each dot is one animal. So there are some variations, of course, but I mean, I did not observe any like dramatic, you know, difference that would suggest there are two subpopulations of animals that respond qualitatively differently. No, I didn't observe that.
And then have you looked at all in how widespread the growth of the spines are? Because I know there's been some work looking at BDNF, for example. I know some of the kind of roadblocks that's happened from that is there's kind of been uncontrollable growth past, you know, what's expected. And I wonder if you've looked at that at all with this.
You mean the BDNF pathway? No, I haven't looked into that. I haven't like applied any manipulations directly on the like a BDNF or its downstream signaling. Yeah, but I mean that is also, you know, one signaling pathway that is quite extensively studied in the context of these synapse formation and plasticity, right? So that's yeah, it is possibly involved in the molecular mechanism of TBG. So you know, that's something we could look into in future.
Thank you. So I have a question from Ryan in the chat, and they're asking, were there any adverse effects that were experienced by any of the mice subjected to TBG?
We didn't observe any like a gross abnormalities behaviorally.
And I think you mentioned before too that it actually seemed to have a lower cardiotoxicity to it, correct, than the ibogaine that was isolated from?
Yeah, that was sorry, let me go back there. So the cardiotoxicity problem is a problem of the ibogaine. So that is actually known even in humans. There were some cases of like a human death that could potentially be attributed to ibogaine cardiotoxicity. But with tabernanthalog, it is largely like abolished or very low. So that was assayed on I think on a zebrafish model in this paper.
Great, thank you so much. And we have another question from Adam, and they say, you mentioned that the hallucinogenic properties of psychedelics can serve as a bit of a barrier in this space. Do you anticipate that efforts to modify compounds like TBG will become commonplace, or are there other examples of modifying psychedelic drugs that you are currently aware of?
Okay, so I know Dr. David Olsen's group is continuing to develop other analogs of psychedelic drugs. So I mean that is, I think, one route that we can pursue. It is actually an interesting thing is that there is still a debate actually recently about like whether the hallucinogenic effects are indeed, whether they are necessary when you are trying to use them to treat certain psychiatric disorders, or they are just side effects that we should try to get rid of. I don't think we have a consensus on that yet. So I think nowadays people are developing these new compounds as well as studying and testing the existing classical psychedelic drugs such as psilocybin and DMT in greater detail. So yeah, I think we need more studies, especially like on human subjects, to really see whether hallucination is beneficial to the treatment or it's something that we should try to remove.
Thank you. And so thank you so much for answering all our questions. Just one last question to wrap up. Mm-hmm. What are kind of the future studies plan for TBG? Where is the future of this research program going?
Well, so as I just mentioned a little earlier, I'm trying to look into the longer term effects of TBG and trying to compare that more with some classical psychedelics and see how that affects the behavior and the brain circuits. I think TBG will, I mean, I'm not sure, but I think Dr. David Olsen is very interested in pushing it more towards like the clinical applications. So that is another direction. I'm sure he would be interested in continuing.
Perfect. Well, thank you so much, Dr. Liu, for a great talk, very well explained. And thank you so much for answering all of our many questions that we had. It was really great to have you on, and I will pass it off to Leo to finish us up.
Thank you.
All right.
Leo, before you start, I just want to jump in really quickly, just to kind of follow up on Adam's question there. So to answer your question, Adam, there are actually companies in the space right now that are trying to commercialize drugs that don't have psychedelic effects. An example of one of those, and it's very topical because we were speaking about ibogaine today, there's a company called MindMed, and they've got a compound called MC18, which is just a different version of ibogaine that's non-hallucinogenic. So great question. Thanks.
Yeah,
Sorry,
and speaking of MindMed, a great segue. Our next meeting is actually going to be February 3rd, so a month from, or a little less than a month from now at the same time, 8 p.m. Eastern time. And our attentive guest speaker is Dan Carlin from MindMed. He is the CMO there. So Hopefully everything works out fine and he'll be able to show up. But otherwise, thank you guys all so much for coming today. I've posted our feedback form in the chat, as well as our email and mailing list if you want to share the Journal Club with anyone. Thank you all so much for coming, and hopefully we'll see you guys all next month. Take care. Bye-bye.
Thank you. Bye-bye.
Thank you, Dr. Liu.